Hey guys, in this video we're going to learn about the sources of genetic variation. So I've been mentioning as we went through the process of meiosis that all of the gametes that we produce are genetically unique. So that means they're not the same as any of the other gametes and they're not the same as the parent cell. So we got to find out, where is this genetic variety coming from? What's making these cells all be unique and all be different to each other and to the parent cell? So there's several different processes that cause the gametes to be unique genetic individuals. So remember, each new human is genetically unique. This is why we can use DNA to match the perpetrators of a crime with the evidence that we got from a crime scene, and we're going to see how that works when we get on to chapter 10 later. But everybody is genetically unique. There's only one exception to this rule: if you have identical twins. Identical twins are kind of special. So the egg and the sperm get together, and then that makes the zygote, right? Remember that? And then usually the zygote multiplies by mitosis to make the embryo, right? But in identical twins, something very special happens. In this very early stages of the embryo development, the embryo splits and it becomes two separate individuals. Excuse me. So with the identical twins, they were made with the same genetic information from the same sperm and the same egg, and they both came from the same zygote. So those two do actually have matching DNA. So just remember that identical twins are the rare exception. Identical twins do have identical DNA. And there's a different type of twins that are called fraternal twins. Fraternal twins, these happen when a female releases 2 eggs in a month. Remember, we said usually a female releases 1 egg during ovulation. In a fraternal twin case, the female releases 1 egg from the left ovary and 1 egg from the right ovary, and then they both get fertilized by different sperm. And then this makes 2 different individuals who have very different DNA. So fraternal twins are twins who are not identical. They have different DNA. But identical twins are the one exception to the rule, and they do have identical DNA. Okay? But remember, generally every human is genetically unique unless you have an identical twin. So we all have a different genome to compare to our mother, our father, and our siblings. So what are the sources for this genetic variation? So here this slide has them all listed. It's a very text-heavy slide, so let's look at some pictures. So one of the first ways that you can make your gamete genetically unique is, remember we said, during prophase 1 of meiosis, the process called crossing over occurs. So crossing over is when homologous chromosomes swap DNA. So they're passing DNA back and forth. So this orange one is the homologous chromosome from the dad, and here is its copy, its sister chromatid. This pink one is the homologous chromosome from the mom, and here's its copy. So notice that they're lining up next to each other. That's called synapsis. And then crossing over, see how they're starting to wrap around each other, and then they swapped a little bit of genetic data with each other. So that's the crossing over part, is when you swap the genetic data. So now the paternal chromosome, the one from the dad, has a little bit of data from the mom's chromosome on it. And then the maternal chromosome from the mom has a little bit of the genes from the dad's chromosome on there. So they swapped genetic data. So it's called crossing over. And when they cross over, they make a cross shape. So the Latin for cross is chiasma. So that's what, where this term comes from. So we'll see a picture coming up from the textbook where they show you a chiasma. So here we've got a pair of homologous chromosomes. This one is from the male parent. This one's from the female parent. This is a copy of the female chromosome. This is a copy of the male chromosome. So remember, these two are sister chromatids, right? And these two will also be sister chromatids. And remember, sister chromatids are joined at the centromere. So remember, during prophase 1, the two chromosomes are going to move close together and line up next to each other. So that's synapsis. And see how they're making an X shape here? That's a chiasma. And when the chiasma forms, then crossing over is going to occur. So notice that they swapped a bit of genetic data here and a bit of genetic data here. So it doesn't have to just happen once. It can happen many times. When they're lined up next to each other, there can be several points of crossover, several places where they exchange DNA. So now is this chromosome, is this sister the same as this sister now after crossing over has occurred? No, they're different to each other. So this is how you get some of the genetic variation. This crossing over process is really important. And then another really important thing to know is, when does it occur? It occurs during prophase 1 of meiosis 1. So here it's just reminding you, crossing over takes place in prophase 1 of meiosis 1, right? So during prophase 1, the homologous chromosomes line up, form crosses with each other, and then they do this process of crossing over. And then once they cross over, then you've got a lot of genetic variety because you're swapping genetic data between the maternal chromosome and the paternal chromosome. Now, another thing that's going to contribute to the increased genetic variety is that the distribution of chromosomes during meiosis is random. So it's completely random how chromosomes are going to line up for meiosis. So here in this picture, you can see that both the blue ones are on the left side, right? And both the red ones are on the right side. So here we got red on the right, blue on the left, blue on the left. But it doesn't have to line up like this. They line up randomly. So you could have a blue on the left, a red on the left, a red on the right, a blue on the right, right? They can line up in any different way. And remember, you have 46 chromosomes. So that gives you a huge variety of the way that the chromosomes can line up during metaphase of meiosis 1. So depending on how they line up here, that's going to give you very different results of which chromosomes are going to be present in your gametes down here, right? The lining up of the chromosomes during metaphase is completely random. There's no way of knowing which one's going to go where, or which pole each chromosome is going to head towards. And then a third reason for genetic variability is that fertilization is random. When a male ejaculates, it releases thousands of sperm in that one ejaculation. And we have the one egg waiting for the sperm, but how do we know which of these guys is going to be the one to fertilize the egg? And actually, you need a high sperm count to fertilize the egg, because the egg actually has a thick protein coating all around the outside. So in order for the sperm to get through, it has to digest this coating that's around the egg. Now, each sperm has a little bit of enzymes in the head of the sperm, but you need a lot of enzyme to digest your way through the coating of the egg to get to the nucleus inside, which is where the DNA is going to be. So you need a high sperm count so you've got enough enzyme so you can digest the wall of the egg to allow the DNA to get in. So we don't know which of these sperm is the one who's ultimately going to make it through to the nucleus of the egg to fertilize the egg. So again, it's completely random which sperm is going to fertilize which egg. So those are some reasons for your genetic variation. So we've got the crossing over, swapping genetic data. We have no way of controlling which pieces of data are going to be swapped, no way of knowing where the X's, the chiasma, are going to occur. So this is a very random process, and it's going to cause an increase in genetic variation. We don't know which way the chromosomes are going to line up during metaphase. They just have to get in line, but we don't know whether the maternal ones or the paternal ones, which directions they're going to be pulled. So there's 8 million possible combinations there for the ways that the chromosomes could line up. And then we don't know which sperm is going to fertilize which egg. So there's 64 trillion combinations from that process. So you can see that by the time you've gone through all three of these processes, that it's extremely unlikely that any one person would have the exact same genetic data as any other person. So like I said, the one exception that we have to this is our identical twins.